Good afternoon, everyone. I hope the lunch coma has passed and you are still with me. Today my name is Ivan. I'm CSO and co-founder of Copenhagen, a startup founded in Sweden. And I'm going to talk about precision genotyping tools for gene editing analysis and characterization. And then I wanted to start the talk today with more of a thought in like we really think that to be able to exploit or to reach the full potential of gene editing, we also need very extremely good validation tools that are accurate, precise, and fast. And it's not only about this, but like I think I'm starting hearing more and more the terms of accessibility, point of care, reliable manufacturing, and it's not only about the gene editing tools, but how we measure how accurate they are, right? Like safety is also a big concern in here. So if we look at in general, like a very general, what is the toolbox that we have to do genotyping for gene editing? We have this. I put together here very general the general methods that we have for this, right? And I put NGS in yellow or gold here because yeah, it's our gold standard, right? It gives us the highest precision, the ultimate resolution. But when we think about things like accessibility and then how really accessible is NGS today, we don't have an NGS as common as having a thermocycler in the lab, right? So, and there are many advances. Illumina and Roche are doing like huge steps towards having this cheaper and more accessible. But until we get there, maybe gene editing is something that really keeps up to the pace because it's going. Also, gene editing is going at huge steps. Every year I come here, things like change drastically. So we at Countagen, we have a technology that is PCR-free and allows for single molecule genotyping. It's a little bit different from what is the conventional PCR-based methods. And in the next 3 slides, I will go a little bit on detail on how does this work. But then, yeah, PCR-free single-cell genotyping, and it's also a microscopy-based readout. So to achieve high accuracy, we use molecular tools that are called padlock probes. These were initially described 20 years ago. And basically, you have a target. In this case, it's a genotype that is the result of a gene editing outcome. So it can be your wild type and your or your expected edited sequence. And then the padlock probe is also a linear oligonucleotide, like the 2 arms are complementary to this target. And in the middle, we have this linker. This linker makes these padlocks very versatile. You can basically load this linker with any sequence you want and tailor it for a different readouts. We are using it to be able to detect or to use microscopy as a readout. But as I will show you in the last slide, this opens many other possibilities. Anyway, so when these 2 arms find the complementary target sequence on the target, then it forms this circular structure. So then you have a hybridization based recognition, but on top of that, we also use a ligase that is highly astringent. So if it finds a mismatch in this ligation site where the phosphate group is, then it will simply not ligate it and not form the circular structure that we will need for the next step. So this process is very stringent, so you need a hybridization event that we do at relatively stringent temperatures and a ligase that has a very good accuracy. So once you have this circle form, then we do another process that is called rolling circle amplification, which basically takes the circle and use the target itself as a primer to start sort of multiplying this target many times. What you end up with is with a kilobase, tens of kilobases long DNA molecule that it doesn't stay as a linear one due to thermodynamic properties. It just sort of collapses in a ball, like this spaghetti that we have there in step 5 called RCA product. And it's a relatively large molecule, so it's around 500 nanometers that we get these very large molecules. And why is this really good? Because it gives large molecules to be able to detect them by fluorescence microscopy and enable this single molecule quantification that gives us very high precision. So I put on this slide something from David Wald to say that the highest resolution measurement one can make is at the single molecule level. It just doesn't get any better than that. And the tools that we are using enable for that. So then now we have this nanoball, DNA nanoball, Roloni, it has been called in literature in many different ways. That has, and this linker has complementary sequence to oligos that are tagged with fluorophores. So then when you hybridize a bunch of fluorophores to that molecule, you will end up with this super high concentration of fluorophores in a very small volume. And that's how you end up with these different dots. And these dots can be quantified later on, but what I wanted to see is like this is of course a pseudo-colored image, like the one that you see within the circle, that each color corresponds to one genotype, to one original padlock that detected one original genotype. And then if you have followed through, you can think that, yes, okay, but if we start from 50 to 100 nanograms of DNA, you only have around, I don't know, 30 molecules of that particular sequence. That's a pretty diluted one, right? So if you look at that into the microscope, you will have this, you will basically just detect 1 or 2 or 3 dots because they are too diluted. So then in Countergen, we also have adapted a very simple yet powerful concept that is basically bringing all these molecules to a single field of view. Basically, you bring everyone together for the picture. And this is very powerful, and we have found out that, or we have quantified this, and it can be equal as having 14 times of PCR. So this allows us to have very good sensitivity, and then to do this single molecule counting, where like we have with the microscope images, and then we measure the intensity and we cut that, and at the end we end up with a quantification that is either 1 or 0. So we have spent the last few years packaging this into a reagent kit for everybody to use, together with a companion analysis software. So then, the idea is that you can achieve single base mismatch specificity thanks to the padlock probes, as I just showed you. It's PCR free, it's a standard workflow, so we really make sure that as soon as you receive it, you can use it. You don't need to spend time optimizing this kit. It can work from 50 nanograms of genomic DNA or 30,000 cells as input. We have now compatibility with human and mouse cell lines, and we have 2 different formats. I think that the things that are here on bold is the things that are new this year. Together with a companion, this analysis software is now cloud-based. So I brought 2 examples of how this GeneAbacus can be a complement or an alternative to the traditional methods. So then this was work that we did together with the Karolinska Institute, with the CRISPR Functional Genomics Karolinska Institute, where we basically wanted to detect these 2 genotypes outcome of a gene editing experiment. So we have this wild type, and then we have this -74 deletion. And then we basically design also a droplet digital PCR assay to detect how efficient that at the pool level and at the clones level. So then when we measure this with PCR, it was telling something like, okay, probably you have your expected specific -74 deletion in like 80-90% of your pool. While when we quantify this with GeneAbacus, it was only 20%. So then we were further into the clone selection stage, and then we also did Sanger on those ones. And then we found out that the very specific -74 deletion was only present in 2 out of the 9 clones that we picked, and that corresponded more to what GeneAbacus was telling us. And then you can see that on the table of the Sanger that there's a lot of -73 deletions, which the ddPCR without proper optimization on it, you wouldn't be able to catch it. So It's one of the cases that we like to highlight because it really shows that the padlocks together with RCA has this single molecule precision, single mismatch precision. Sorry, and then today we're also bringing today to this conference another use case. So we were approached by a cell line engineering company that works with very limited amount of cells. It's a single cell engineering company, and then they really needed to start from very limited amount of cells. So then the current workflow was NGS based, and as it was a service, it's an outsourced genotyping NGS. And then you usually start from the crude extracts. You need to wait the cells to grow until 10,000, up to 10,000, and then wait another 2 weeks to be able to get the results from NGS. They come to us and then see if we could sort of work out a solution or like to help them really reduce this initial genotyping to really the minimal time. So then we developed something that is cell boost, that is a sample prep module for starting from very small amount of cells or very small input DNA. So we get the most out of this 100 cell over the limited DNA amount, still PCR free, and then you can get genotyping results 2 days after. In collaboration with them, they also really want us to sort of test our analytical performance, and basically they provide us with a wild type and a clone of a -1 deletion. And they prepared basically a dilution series on these, and then basically tested Genavacus. We got an excellent correlation against what was expected. More on this story is in poster 100. I don't know if you already met Hamid last night. There's still time today. The poster is hanging up there, so I invite you guys to hear more about the story. There is more to it. After that, we also tested some blind samples. So then, I invite you guys to really pass by the poster. And then something that is an announcement and something that is new for this year is that before we had Genavacus only to be able to run 8 samples at a time. So it was a bit of a small scale assay. But we have worked really hard last year to make this possible in 96 well plates, so you can do a larger high throughput samples with this assay. So then for this, we are now running an early access program with on-site demos, and then I invite you to pass by booth E3, where Ali can tell you more about this. It's only the first booth when you enter to the right. And then finally, I wanted to bring something new, and it's like, remember that I mentioned at the beginning that the padlocks have this linker that we can basically tailor and customize with. We can load it with sequences for different things, and this includes like things like UMIs, barcodes, sequencing, moieties, and different things. So, and there is another variation of these padlock proofs where you don't need the 2 arms to hybridize next to each other, but you can leave a gap in between and have a polymerase fill in that gap, and then the ligase comes again and closes the circle, and then after that is a similar process of as I described. So then we basically generated a proof of concept of this for detecting a heterozygous clone, also using the amino ion sequencer. And this is a very early stage. This is very like from the oven that we are able to do this. But my message here is I really wanted to tell you like the potential of this technology. Like right now, we are working with Genavacus doing this sort of complementary technology to make the whole genotyping faster. But we can also just bring it, push it forward to be able to do indel profiling and more. And then with that, I finish. I really thank you today for giving me the opportunity to be here. Visit our booth, visit our poster. We are around. Like half of the team is around, so you can meet us. I will be happy to discuss with you and chat about PCR challenges, or how can we really bring, how can our technology help you with genotyping in general.